Hello everyone. Today we are going to learn about serial dilution techniques for cell counting. Okay. So before we get started, the first thing we need to know is that who discovered it, right? So Robert Koch is the scientist who discovered this serial dilution technique. This is Robert Koch, as you can see, and he is the one who is credited for identifying method that is used for bacterial enumeration. Now he first did this method, applied this method on the water sample. Okay, he also wrote an article about it. The name is About Detection Methods of Microorganisms in Water, and this article was published in 1883. Now, the standard plate count method using serial dilution is a reliable method for enumerating bacteria and fungi. Now, this common practice is not only used for liquid but also used for solid specimen. It is also useful to count bacteria, fungi as well as virus. Okay. So, what is the importance of the serial dilution? Now, as we know that microorganisms are present in soil, air, water, everywhere, and they are in high numbers, right? So, the exact number could not be found out because discrete or separate colonies are not growing in the medium. If you take if you do the sampling from the original sample, right? So, it is not possible to count it. So, this serial dilution method, what we do in this method, the microorganisms are grown here, and after doing the serial dilution, this microorganism, they grow separately. All the live propagules will grow and develop into an individual colonies, which will allow us to count them easily. Okay, so what happens in this method is that a known volume of the soil or water sample is suspended into a known volume of the sterile distilled water. Then, known volume of the suspension, like 1 ml, is transferred from the stock to the additional flask, which contains 9 ml, to get serially diluted suspension of 10 to the power minus 2, 10 to the power minus 3, minus 4, minus 5, and so on. So, from there, aliquot of 0.1 ml suspension is poured on the surface of the medium which we have prepared, and this medium is then incubated at a given temperature for a given time, and the colonies are counted based on the serial dilution plate number. Okay, so this is the importance of the serial dilution because it helps to grow the each microorganism into a separate colony, which makes it easier for us to count. Now, coming to the requirements that is needed for serial dilution. So first we need a culture media. It may be nutrient, agar, luria, buttony media, whichever media you want to take, you can take it. Second is the sample we need. That is you can take either solid sample like soil or liquid sample like water. We need a flask, usually 50 ml or 100 ml, test tubes, pipettes, petri plates, incubator, autoclave, distilled water, and spreader. Okay. Besides this, you also need to have a gloves. It's better to use gloves when you're doing microbial work. Okay. So these are the requirements. Now coming to the procedure of the serial dilution. So what we do in first, that is we prepare the required agar media, that is the culture media, and we autoclave it at 121 degrees Celsius 15 lbs for 15 to 20 minutes. Now in step 2 we take a clean Erlenmeyer flask and pour 10 ml distilled water to it. Okay. Now remember that you need to heat sterilize the glassware before use. And the distilled water which you are taking for the serial dilution should be autoclaved as well. Okay, so this is the note you need to remember. Now in step 3 we measure 1 gram of the soil sample and we add it to the 10 ml distilled water containing flask. Then we mix it well, and this sample is called as the stock soil sample, and you label it as 10 to the power minus 1. Then after that you take 5 to 6 flasks more and add 9 ml distilled water to each. Then you label it as 10 to the power minus 2, minus 3, minus 4, minus 5, minus 6, minus 7 till label it till you want the dilution. Like if you want to do it to 10 to the power minus 6, label it till that, and you can increase the dilution as per your requirements. Now after that you mix the soil sample properly in the stock solution, that is 10 to the power minus 1, and you take 1 ml from it and add it to the second flask which is labeled as 10 to the power minus 2, which contains 9 ml distilled water. So here the total volume will become 10 ml. And this process will continue till you reach your last flask. Okay. So here it will become more clear if you look at the examples here. So this is the soil sample which you have. Now you take 1 gram of the soil sample and you give it to the 10 ml distilled water. Okay, and you label this one as 10 to the power minus 1. This is the stock soil sample which you have. Now here the total volume is 10 ml. Okay, now from here you take 1 ml and give it to the second one, which contains 9 ml of the distilled water and is labeled as 10 to the power minus 2. Okay, so here also the total volume will become 10 ml. Now the same process is continued. You take 1 ml from the second one and put it to the 9 ml distilled water in the third one. Now this is labeled as 10 to the power minus 3. And this process will continue till you reach your last dilution. Okay, you take 1 ml from the first one and put it in the second one. You continue it till you reach your last dilution. So here we have taken till 10 to the power minus 6. Okay, so this is how the serial dilution is done. Now after the serial dilution, you need to plate it, right? You need to grow these microorganisms on your plate, on your petri plate, having the culture media. So how you do it? Now from coming to the plating the dilution sample. Okay, now this petri plates which you have autoclaved with culture media, you pour the agar media in the laminar and allow it to cool for 20 to 30 minutes. Now take the dilution flask of your interest, usually the last flask, 10 to the power minus 6 or minus 7. Here we have taken 10 to the power minus 6. Okay, so take 0.1 ml from it using a pipette and add it to the plate. Now after adding, you spread the sample evenly on the agar plate till it gets absorbed completely. Now label the petri plate properly so that you can identify which plate is of which dilution, right? Now what you do, you keep the plate in the incubator at a given temperature for a specific period of time. Generally all the microorganisms grow at 37 degrees Celsius and they require incubation period of like 24 to 48 hours. Okay, so this is the general temperature and time period. Now we need to remember that temperature, pH and time of incubations are different for different organisms. Okay, so different microorganisms grow differently. So you need to remember this. After that, you observe the colony on the plate next day and count the number of colonies that you have on your plate. Okay, so if you see this image, this is the soil sample which we have. We have taken 10 to the power minus 6 dilution. What you do, you take 0.1 ml from this solution, you put it on the agar plate, and then you use a spreader to spread it properly on the media till it gets absorbed completely, and then you put the plate in the incubator at a given temperature and time, and after that you will observe the colonies. Okay, so here you can see that the colonies are separated. Okay, separate colonies here you can see, and this is countable. Okay, so why are we taking the last dilution basically? Okay, so what happens is that if you take 10 to the power minus 1 dilution and you plate it, you will find a lawn of microbes that is not countable at all. Okay, and if you take the 10 to the power minus 2, still it is uncountable. As you can see, there is so many number of microorganisms. In 10 to the power minus 3 also it is uncountable. 10 to the power minus 4, from 10 to the power minus 4 it is getting little countable. 10 to the power minus 5 is countable and 10 to the power minus 6 is easily countable. So you take that dilution where you can find the colonies countable. Okay, so usually countable colonies are taken, those countable colonies are taken which are between 25 to 250. Okay, so this is why the last dilution is taken. Now, what if we take a liquid sample? Okay, so the procedure for the liquid sample, for serial diluting a liquid sample is you take the required agar media, prepare it and autoclave it. Then you take a clean test tube and pour 9 ml distilled water to it. Okay, Always heat sterilize the glasswares before use, and distilled water should be autoclaved as we studied in the serial dilution of the solid sample. Okay, so here you take the 1 ml of the water sample which you have collected from the original source and you mix it in the 9 ml distilled water. So this is called as the stock water sample. You label this one as 10 to the power minus 1. Now after that you take 5 to 6 test tube more and add 9 ml distilled water to each one. Then label it as 10 to the power minus 2, 10 to the power minus 3, minus 4, minus 5, minus 6, minus 7. Next what you do, you mix the water sample properly in 10 to the power minus 1. And from there you take 1 ml and add it to the second test tube which is labeled as 10 to the power minus 2, which contains 9 ml of distilled water. Okay, and the total volume will become 10 ml. Now after that you continue this process till you reach your last dilution, that is 10 to the power minus 6 or minus 7. Okay, so this will become more clear if you look at this pictorial presentation. So this is the water sample which you have collected from the original source. You take 1 ml of this water sample and add it to the 9 ml of distilled water in the first test tube, which is labeled as 10 to the power minus 1. So this is the stock water sample which you have. Now you mix it properly, and then what you do, you take 1 ml from this first one and put it on the second one which contains 9 ml. So here also the total volume becomes 10 ml. Okay, and you go on with this process till you reach your last dilution. Okay, so this 10 to the power minus 4, you do it for minus 5 as well till you reach 10 to the power minus 6. Okay, so usually the countable number of colonies appear between 10 to the power minus 4 to 10 to the power minus 6 dilution. Okay, so the process is similar to that of the solid sample. The only thing that is different is that in the solid sample we are taking 1 gram in the first one, 1 gram solid sample, and we were adding it to the 10 ml of the distilled water here in the first one, 10 to the power minus 1, whereas in the water sample we are taking 1 ml and adding it to the 9 ml, so that the total volume becomes 10. Okay, so this is the sole difference which we have between these two dilution method. Rest all the methods are same. Now you need to do the plating of your dilution sample, right? So the process is same as the previous one. The petri plates are poured with agar media in the laminar and allowed to cool for 20 to 30 minutes. Then you take the dilution test tube of your interest, that is 10 to the power minus 6, and take 0.1 ml from it using a pipette and add it to the plate. Now after adding it, you spread the sample evenly on the agar plate till it gets absorbed completely, and you label it properly after that. Then you keep the plate in the incubator for the given temperature and time, and observe the colony formation on the next day and count the number of colonies. Okay, so this process is similar. You take the water sample, the dilution of your interest, that is here we have taken 10 to the power minus 6. You take 0.1 ml from this sample and add it to the plate, and after adding you spread it evenly on the plate, and after it gets absorbed completely you put it in the incubator. Then after that you will find that the colonies have grown separately, which is countable. Okay, so the same process is done here. Why we take the last dilution sample? I have already told it in the soil sample. So if you take the 10 to the power minus 1, you will get a ln of micros which is not countable. Then as you go on increasing your dilution number, you will find that the number of colonies are getting decreased. Okay, so you take that dilution sample which will give the colony number between 25 to 250. Okay, so this is why the last dilution is taken for the counting method. Now from here you can count, not only count the number of colonies in the one dilution sample, that is 10 to the power minus 6, but you can also find the number of colonies that was present in the original soil sample from the soil sample or the water sample from which you are taking it. Okay, so the original sample also you can find it. Now the observation which we'll have after growing it in the agar plate, we'll find a small shiny bacterial colonies that is grown on the agar plates, like as shown in this picture. Now fungal colonies will obviously have different size and color, as you can see, and small radiating colonies of powdery appearance will come for actinomycetes. So this is how the bacterial colonies will occur. Now if you want to learn more about the different types of microbial colonies, you can comment it below and we'll make another video on it. Okay, so this is what we'll observe. Now for CFU counting we have a formula. Now this formula is divided into two parts. First, you need to determine the concentration of the cells that is present in the diluted sample. So here the diluted sample which we have taken is 10 to the power minus 6, right? So if you want to count it, you have to follow this formula. So CFU in the diluted sample cells per ml is equal to number of colonies that is counted on the Petri plate divided by the amount of diluted sample that is added to the plate in ml. Okay. And the second step is determination of the concentration of the cells in the original sample. So CFU in the original sample cells per ml can be found using CFU in the diluted sample divided by dilution of Petri plate counter. Okay, so this is how the total CFU counting can be done for the diluted sample and also for the original sample. This CFU counting, details on the CFU counting is given in our CFU counting video. You can go and watch it if you want to know about the CFU counting in details. In that video, it is explained with examples also. Okay, link is in the description. You can go and check it out. So this is all for today's video. Hope you have understood it. If you have any queries related to this, you can comment it below. If you want a detailed notes of this video, you can follow our LinkedIn page. The link is in the description as well. And do like, share and subscribe to our channel for more such related microbiology updates. Thanks for watching.